Hello everyone, I am Dineka Tilakaranna from Sri Lanka and today I am going to share one of my research, that is the contribution of terrestrial crabs on organic carbon sedimentation in mangrove ecosystem. So before we move on to this presentation, I would like to ask you a question. So what are mangroves and what are the factors that affecting for this carbon sedimentation process? We will look at one by one. So mangroves are the ecosystem which are closely located in the coastal belts. So in this map, you can see these mangroves are highly located in the tropics and temperate regions. And they are highly efficient at the carbon sequestration. So they are mainly considered as the globally important carbon sinks. So how they are act as this carbon sink? Basically, they use biological and physiological processes in these carbon sedimentation processes. What are the biological factors involved in this carbon sedimentation process? Crabs. Crabs are the biological factors who are responsible for this carbon sedimentation process. These terrestrial crabs are highly abundant in this mangrove ecosystem. They play an important role in recycling of the carbon. However, their contribution is not yet studied intensively. Therefore, we conducted this research to quantify the contribution of crabs together with other macrobenthos for the process of carbon sinking in the mangrove ecosystem. This survey was conducted in the southern part of Sri Lanka. So in this map, you can see this is the intensive red rod network which are connected to the human settlements, and we established 4 locations closer to this rod network. We consider these plots as disturbed sites, and inside the dense forest, we again establish another 4 plots. These 4 plots were considered as the least disturbed sites for our study. This study was conducted for about a 1-year period. We established 8-10 square meter plots for each location. We calculated the vegetation composition and the stem density within these 100 square meter plots. Then we took the crab abundance as well as the crab hole abundance while establishing a 10 square meter subplots inside these main plots using these kind of quadrants. And crab biomass is weighted using a digital balance, and size of the crabs, basically the carapace width and the length and the size of the crab holes were measured using a vernier caliper. Abundance of the macrobenthos was estimated using a 500 cubic centimeter soil augers. For the abiotic component, we calculated the soil pH, soil salinity, as well as the soil moisture content using a standard methods. For the data analysis, we calculated PCA to analyze the effect of physiochemical parameters on the study sites, and per manova to test the significance of species abundance and amount of standing stocks between disturbs and these disturbed sites. The Spearman's rank correlation to test the correlation of crab abundance with soil parameters. Finally, the linear regression models were run to determine the appropriate relationships of soil organic carbon with the stem density, standing stocks, and the abundance of the crabs. During this survey, we were able to encounter 3 crabs from this mangrove ecosystem. So they are the main contributors for this biological process of carbon sedimentation in the mangrove ecosystem in Sri Lanka. Neosomathium smithi, Episocerma versicola, and Perisocerma glutinatum was those 3 species. Out of these 3, we found that this Neosomatha smithi was the largest species. So it has the carapace length of 2.46 centimeters, and the smallest one is the Perisocerma species. It has the carapace length of 1.99 centimeters. Interestingly, we found that these crab hole structures are species specific. So in this diagram, you can see the three different species have three different structures of their crab holes. Not only that, these crab hole structure as well as the diameter of these crab holes are highly correlated with the amount of soil carbon sedimentation. So we found that the biggest species have the biggest hole size in this ecosystem, and they are the most efficient and the highest contribution for this carbon sedimentation process. We found that soil organic carbon was vary between 8 to 19 kilograms per 1 square meter plots inside this ecosystem. In addition, we found that positive relationships between soil organic carbon and standing stocks, crab biomass, crab abundance, and stem density. They are increased with respect to each of these factors. So this, when we consider in this crab biomass and crab abundance, when the crab biomass and the abundance getting increased, they are highly efficient at sinking of carbon. Because we clearly observe that once the fallen leaves or propagules or the seeds of these mangroves onto the grounds, they are quickly come out from their holes and taken those live matters inside to their holes. Then they are digesting those things and they again out those digested materials out of their burrows. So they are highly efficient at carbon sedimentation process. In addition, we want to look at what are the most abundant burrowing materials inside these crabs' holes. So we found that shells are the highly abundant burrowing materials inside these crab holes, and it followed by propagules and seeds of non-mangrove species. Then we want to look at how these sampling locations were clumped in a three-dimensional space. Therefore, we conducted the PCA analysis and we found that site 1, 2, 4, and 3 were clumped together. So these are the disturbed sites, and site 5, 6, 7, and 8 were clumped together. Those are the least disturbed sites. So these two sites are separated due to the soil pH and soil conductivity. So in these two photographs, I want to highlight that this is the disturbed sites where our disturbed locations were established, and this one is the dense mangrove forest. We established our least disturbed sites. So this is the road network connected the human settlements, and beside this road network, we established these disturbed plots. And we found that abundance of these crabs in these disturbed plots were relatively low compared to the dense forest because of the anthropogenic activities and the anthropogenic disturbance. Then we want to look at how crab biomass and soil carbon is vary between these disturbed and these disturbed sites. So we plot this box plot to get an idea, and we found that the crab biomass as well as the soil organic carbon is least abundant in the disturbed sites compared to the least disturbed sites. It's mainly because of the low number of crabs were abundant in the disturbed site due to the human activities. Then we want to look at how these physiochemical parameters vary between these sites. And we found that soil pH in the disturbed site was less compared to the least disturbed sites, and conductivity, soil salinity, and soil moisture was higher in the disturbed sites compared to the least disturbed sites. So these are the physiochemical parameters that affects for the abundance of the crabs in this ecosystem. So this abundance of this crab then ultimately relate with the soil sedimentation process. At last but not least, These crabs have preferentially live under the dense mangrove vegetation and they act as ecosystem engineers. So they modify their soil character, especially the soil water holding capacity, moisture content, soil organic matter content. So those kind of factors they are modifying. And this nutrient transformation into the deep soil was mainly conducted by these crabs. Once these leaves and the live matters were placed on this ground, they taken those live matters into their burrows and they help to transform these nutrients from top of the soil to the deep of the soil, and they enhance this long-term preservation of this soil organic carbon process. Therefore, this mangrove ecosystem is act as a efficient carbon reservoirs. And on the other hand, the conservation importance of this mangrove ecosystem as well as the conservation importance of this population of these crabs is really important for the carbon sequestration process on the earth to balance the carbon between air and the land. So these are my references, and I would like to thank for the Ministry of Environment of Sri Lanka for their funding support and ATBC for giving me such a great opportunity to present our research work, and thank you for your patience.